Welcome to the next lecture on microbial ecosystem and biogeochemical cycling. In this lecture, we are going to discuss about the concept of guilds formed within a microbial community or within an ecosystem, and how the formation of guilds by different members of the community actually facilitate the workings of the ecosystem. And during this deliberation, we are also going to highlight the interrelations between the different microbial populations that actually form the basis of the guild, and the guilds merge into communities, and the communities actually facilitate the workings of the ecosystem. We are also going to discuss the microorganisms and microenvironment. So, in particular, how microorganisms play a role within different microenvironments that present in any habitat will be emphasized, and finally, we will proceed towards the biogeochemical cycling. So, we begin with the basic concept of how the different microbial populations interact to form the guild and the community functions towards the ecosystem, the broader working or the broader ecosystem goals or functions that are catered by the communities. So, as you can understand that in any ecosystem or within ecosystem, we have multiple habitats, or within the habitats we have numerous microbial populations. So, as for example, we have actually taken 4 populations. So, these populations like A, B, C, D are marked, and each of these populations are actually represented by a group of microbial organisms which belong to same species. So, population A is actually a member of all members of a particular species, population B another species, population C another species, and population D is another species. So, 4 species are taken as an example. Now, metabolically linked or metabolically related populations, they form guild. So, guild is kind of a representation where microbial functional assemblage is indicated or discussed. So, a group of populations that may be more than 2 or more than 3 or more than 4. For simplicity, we have considered 2, but it can be any number of populations, but they are definitely distinct populations, and each of these populations are having some kind of metabolic relatedness or interconnectedness. So, they basically share certain metabolic functions, and they perform certain metabolic functions together. This metabolic linkage forms the basis of guild. So, in this case, the population A and population B forms the guild 1, and population C and population D forms the guild 2. Now, there could be a large number of guilds, as we can understand, within a particular environment or within a particular habitat, and all these guilds they represent the community function. So, the guilds are basically the metabolic linkage among the populations, and together these guilds they represent the community. Now, this is the microbial community. Now, this microbial community present in any habitat or any ecosystem continuously interacts with the abiotic factors that we have learned earlier, and it is not only the abiotic factor and the microbial community interaction, but it is also the interaction between the macroorganisms, the eukaryotic organisms mostly. This interaction continues, it is a kind of a 3-way interaction between these community, microbial community, macroorganisms and abiotic factor, and that ultimately leads to the workings or the forms the basis of the workings of the ecosystem. So, whenever we are working on any kind of environment ecosystem for maybe for energy recovery, maybe for carbon sequestration, maybe for any kind of issues, environmental biotechnology issues related to climate change, removal of a particular pollutant, remediation of any specific pollutants. So, we must need to understand the workings of the ecosystem, which is fundamentally catalyzed by these interactions, and the interactions are again facilitated by a multi-layered structure of these populations and their activities. Now, this linkage, that is the linkage between the multiple populations present forming the guilds to communities. So, in a habitable ecosystem whose resources and growth conditions are suitable, That means the conditions are favorable, the nutrients are available to the organisms. Individual microbial cells present there will grow to form the population. So, that is a kind of inevitable. So, if you have sufficient nutrient resources, sufficient or favorable growth conditions, obviously the microorganism species who are present there will grow and proliferate, and that proliferation will lead to a formation of different cluster of population. So, we can see there are 3 populations which actually have grown into this into the soil, where plant a fraction of the plant root is also there. Now, these metabolically similar populations, for example, as we indicated earlier, population A and population B, for example, that exploit the same resources. Like, for example, in this case I have considered nitrate as the electron acceptor for maybe these 2 populations, the green population and the yellow population. These 2 are 2 populations. These populations are capable of, both of them are capable of denitrification. So, they are capable of reducing nitrate to nitrogen. So, they form a guild. So, we can consider that the metabolic linkage between, or metabolic closeness, metabolic association between these 2 populations are forming the guild, and there could be several such guilds present within the community, and all these guilds together represent the community, and the communities interact with the, as I mentioned earlier, with the other organisms and the abiotic factors, and that is actually going to define the working of the ecosystem. Now, this can be very well explained using a surface water ecosystem, where we will see also not only the association of different metabolically linked or metabolically associated or metabolically connected microbial populations, each of them are represented by a particular species, but also we will see how the flow of energy, flow of nutrients are allowed, and we also will see how that facilitates the cycling of the nutrients, which forms the basis of the biogeochemical cycle within any environment. Now, energy flow and nutrient cycling in any ecosystem are achieved through the coordinated function of various populations. So, remember, these populations we are referring to actually the microbial populations, which are represented by numerous species. So, each population is basically a specific species, and these populations group together to form the guilds and the community. So, the energy flow and nutrient cycling are achieved as the populations interact or interlink through their metabolic relatedness, and they form the guilds, and the guilds form the communities. Now, before we go into this one surface water system and try to understand how actually these functions, we need to understand that in any kind of ecosystem, the major microbially driven processes can be categorized into 3 major. There are many ways that we can actually categorize the microbial activities within any ecosystem, but for our simplicity at this moment of time, we are considering 3 main microbially driven processes, which are the phototrophy, that is using the solar energy as the source of energy. The microorganisms are capable of their growth and nutrition. They fix the atmospheric carbon dioxide into organic matter. Chemoorganotrophy, that means the chemical energy is used and the atmospheric carbon dioxide is used to convert the carbon into organic matter. And these organisms, chemoautotrophy, could be chemolithoautotrophy as well, because the electron source could be inorganic materials, and organotrophy, that means organic carbon is used as the source of electron, source of energy. So, we can actually categorize very simply categorize the microbially driven processes into these 3: phototrophy, chemotrophy and organotrophy. Now, within each of these ecosystem, or in most of the ecosystems or most of the environments, whether it is the pristine environment, whether it is a contaminated environment, we see there are 3 important other inputs or sources for driving the entire flow of nutrients and energy through microbially catalyzed reactions. So, first and foremost is the energy inputs. The energy may come from the sunlight, energy may come from the oxidation of organic matter and oxidation of other reduced inorganic substances. The second one is the carbon inputs, that is the carbon sources and the extraction of the carbon and its assimilation into the cellular molecules. And this source of carbon could be carbon dioxide, could be organic carbon and other reduced inorganic carbon could also be used. Electron sources include the reduced organic and inorganic substances. So, we are going to discuss in detail in our subsequent lectures that how these are actually facilitated and how they are connected to the environmental biotechnology related processes. Now, apart from these three inputs or sources, two complementary roles of microorganisms can be clearly identified in any of these ecosystems or any ecosystem habitats. So, one is the synthesis of new organic carbon from the carbon dioxide and using other inorganic substances. This is the primary producers. So, mostly in case of microbial ecosystem, as we understand that higher plants or green algae might be there or might not be there. So, some of the ecosystem could be solely populated by bacteria and archaea, but still there could be many bacteria who are chemoautotrophic bacteria or could be photoautotrophic bacteria. There could be surely many algae, photosynthetic algae or photosynthetic other bacteria, but there could be many places where we do not see any kind of photosynthetic or photoautotrophic organisms, particularly in the subsurface environment where there is no solar energy or sunlight cannot penetrate those habitats. So, anyway, so the two complementary roles that are played by microorganisms, number one is the synthesis of organic carbon, that is the assimilation of inorganic carbon into organic carbon, and along with organic carbon lot of nutrients are also assimilated. So, as the cell grows utilizing the carbon dioxide and energy and inorganic nutrients available in its vicinity or in its environment, it grows in number, it creates lot of biomass, and within this biomass this carbon is assimilated as organic carbon, and along with the organic carbon the other nutrients, inorganic nutrients are also assimilated. They represent these type of organisms which are basically the autotrophic organisms, mostly for surface environment these are the photoautotrophic microorganisms, or for different other subsurface or other environments where solar light or sunlight cannot penetrate or sunlight is not available, many of the cave environment or something like that, we see that chemoautotrophs they predominate. So, they are the primary producers. The second type of organisms and their role is basically the oxidation and decomposition of the organic carbon which is available there. This may be because of the dead microbial cells or the organic matter released by different microbial cells during the course of their metabolism. So, this decomposition is facilitated by the oxidation of the organic carbon and release of energy, carbon and other nutrients which are subsequently utilized by other species, other members who are present in the this particular habitat. Now, let us look into this surface water community. So, this is kind of a pictorial image which depicts a surface water scenario, and you can see that there is a water column and the above, and we have also the sediment which is rich in inorganic and organic materials naturally. So, this could be a lake or this could be a river or this could be a pond. So, it is one of the simplest way of explaining how these populations interact to form guilds. Guilds are forming the entire part of the community, and this association between the population to community drives different type of microbially catalyzed reactions, facilitating the nutrient cycling or biogeochemical cycling. So, to begin with, in this system for example, say energy could be obtained from sunlight as the surface is exposed to the outer atmosphere. Energy could also be obtained from the organic carbon, and it could also be obtained from the reduced organic or inorganic substances. Now, these inorganic substances, the reduced inorganic substances are often present as a part of the sediment or as a part of the deposition. The organic carbon is naturally it could be available in the sediment, or it could be also present as a kind of an external source, or it could be generated in situ that is the microorganisms and other organisms, the phototrophic organisms. So, when they grow within this ecosystem, they produce the organic the biomass, as I mentioned earlier, and that biomass could actually form the organic carbon or provide the organic carbon. The cells after their death, the cell mass is utilized, decomposed, and also the different metabolites which are released by functionally active cells are often also contribute to this organic carbon pool. Now, as I mentioned, the energy could be the energy inputs in this ecosystem could be from the solar system. It could also be the inorganic material or the oxidation of organic carbon. So, with the exposure to the sunlight, the first zone that we can identify in this kind of ecosystem is the top layer where the sunlight is available, that is called the photic zone. Now, within photic zone, it is obvious that the sunlight will be utilized by the microorganisms present over there, and naturally the phototrophic organisms who are the photoautotroph, who are capable of utilizing the carbon dioxide present in the atmosphere or the dissolved in the water column, will be utilized in the form of bicarbonate, will be assimilated inside the cell and then will be utilized through carbon fixation pathway. And along with the inorganic carbon, the nutrient will be required. So, inorganic nutrients will be also assimilated like nitrogen, phosphorus, sulfur, iron, etc., which are often present as soluble form within the water column. So, the phototrophic organisms will be capable of converting this carbon into the essential energy form, that is the ATP, and also new organic matter will be synthesized. Now, this new organic matter, as we discussed earlier, will contribute to the organic matter pool of this ecosystem, which can be the cell mass itself or the released metabolites or the metabolic products from these phototrophic organisms. Now, this organic matter which is synthesized, so we call new organic matter, in situ synthesized new organic matter by the phototrophs, actually represent the bulk carbon, and along with carbon, as I mentioned, it will also have lots of nitrogen, phosphorus, sulfur, iron, magnesium, potassium, etc., which are assimilated within the biomass. So, as the biomass is available or the metabolic products are available into the broader pool of nutrients, these will also be available to the system. So, any microorganism like to use them would possibly like to use them. So, in addition to this new organic matter, so there would be some other organic matter sources are also there. So, let us now proceed. So, along with the photic zone, the immediate vicinity will be the oxic zone. The photic zone in fact is also the oxic zone, because the concentration of the oxygen, the atmospheric oxygen will be higher, and of course, there will be a gradient within this oxic zone, because as we go deeper into the water column, the level of oxygen will be declined. It is also a fact that the utilization of oxygen by aerobic microorganisms will further reduce the oxygen level. So, the oxic zone might have a gradient within itself, but for our simplicity we are not considering the gradient at this moment of time. We are just considering that along with the photic zone there is the oxic zone. Now, within this oxic zone there are certain very important activities from the microbial point of view. So, as we have learned earlier that the organic matter will be synthesized by the phototrophic organisms, that corresponds to the new organic matter. Along with the new organic matter, externally derived or leached organic matter can also contribute into the organic matter, organic carbon pool of this ecosystem. So, that is actually called allochthonous organic matter. Allochthonous means these are externally derived organic matter, which could be the runoff, agricultural runoff, or any kind of other sources, maybe from the sediment deposits. These will be leaching into the or mobilized or solubilized, and then they are coming into this water system. So, together these will represent the total organic carbon or organic matter present in the system. Now, within the oxic zone, it is expected that this organic carbon which is present there will fuel the growth of heterotrophic microorganisms. So, heterotrophic bacteria, heterotrophic other archaea would metabolize the organic carbon, no matter whether it is autochthonous or allochthonous. Autochthonous means in situ produce, or the allochthonous, that is externally derived. This organic carbon will be metabolized through microbial respiration, because oxygen is there. So, using oxygen as terminal electron acceptor, aerobic organisms will respire, or in presence of different other terminal electron acceptor, some of the organisms may also grow if oxygen is not sufficiently available at some point of time. So, basically the respiration, and under some situation we see that along with respiration, or in some, for some bacteria, fermentation can also proceed, because if there is lack of oxygen or lack of suitable electron acceptor, or some microorganisms prefer to do fermentation. So, basically the fermentation of the reduced substances like glucose is fermented to lactic acid or acetic acid or something like that, that can also be produced. Otherwise, the organic carbon will be metabolized to carbon dioxide. Now, here we should also include that the organic carbon which is actually utilized by these heterotrophic organisms are obtained from the phototrophic bacteria or the phototrophs, including the algae. It could be or most likely external organic matter will also be there, but in addition to this phototrophic bacteria, there are likely to be chemolithotrophic bacteria. These chemolithotrophic bacteria utilizes different reduced inorganic material and then try to oxidize them, like hydrogen, Fe2 plus, H2S, ammonia, nitrite, etc. as electron donor. So, these are also naturally available. Okay. Hydrogen might be available because of many of the fermentative reactions produce hydrogen. Fe sulfides, ammonia, nitrite, these are naturally available because of the products of the microbial activities, or often they are also present in the sediment. So, essentially, or they may enter into the system from the external sources. So, essentially chemolithoautotrophic microorganisms, they are also contributing to the new organic matter. So, essentially the organic matter which is present in the system is going to be oxidized and by these heterotrophic organism, leading to the formation of the carbon dioxide or some of the fermentative products. Now, we can actually designate these two groups of populations. One could represent the community A, where many of the phototrophic organisms could be there, or autotrophic organisms are there with different sets of populations like chemolithotrophic populations and photoautotrophic populations, and within photoautotroph we can have again different types of populations. Together they might represent the community A. The heterotrophic organisms could be of different, represented by different populations, together they form the community B. Now, we move forward that below the oxic zone, it is expected that the anoxic zone is going to prevail. So, anoxic zone means it is oxygen free zone, because of the diffusional barrier and also the use or the utilization of the dissolved oxygen by the microbial activities, the oxygen level will decline, and essentially this is going to be the anoxic zone or oxygen free zone. Now, within this oxygen free zone, we can clearly identify a number of guilds, and all these guilds which are indicated here are actually represented or facilitated by a group of populations. As we know that each of the guilds must be represented by number of populations, and together they represent the community C. So, together altogether we can identify that in this aquatic ecosystem we have the community A, community B and community C for example. So, there could be actually numerous other communities also present, but generally we can broadly and easily categorize these three communities. Now, for example, the community C in this case is represented or constituted by three guilds. The guild A or guild 1 is the fermentative bacteria, which are responsible for utilizing the organic carbon and fermenting it to acetate, lactate, formate, etc. under the anoxic condition, because oxygen is not there. Guild 2 could be represented by the denitrifying or the iron reducing bacteria, who will use nitrate as a terminal electron acceptor, forming ammonia or nitrogen from nitrate, or will reduce the Fe3 plus as their terminal electron acceptor and will produce Fe2 plus. Guild 3 will be represented by the organisms or the populations who are responsible for sulfate reduction. Sulfate reductions, sulfate will be reduced to sulfite under anaerobic condition, many of these anaerobic bacteria, they are capable of utilizing sulphate as their terminal electron acceptor. And lastly, the guild 4 would be the methanogens. These are basically the archaea present over there. These archaea could be acetoclastic or they could be hydrogenotrophic or hydrogen utilizing archaea, or could be acetate utilizing archaea, and some of them could also help in acetogenesis. So, these 4 guilds could be easily expected. Now, in addition to these 4 guilds, there could be some other guilds represented by organisms who are capable of oxidizing methane. The methane which is going to be produced under this anoxic environment will be available as a carbon source. So, there would be some organisms, both archaea and bacteria, who would be capable of utilizing or oxidizing rather methane. So, these are methane oxidizing bacteria or methane oxidizing archaea. So, essentially methane can be oxidized to C1, different type of C1 compounds, and then C1 compounds will be again used as a organic carbon going to the organic pool. So, essentially we can see that the carbon which actually derived from maybe from the atmospheric carbon or the dissolved inorganic carbon form the organic matter finally trickle down to the formation of methane or so, and then methane can essentially be converted again, or the acetate can essentially be converted again towards the carbon dioxide, and that carbon dioxide can again be recycled within this system. So, we are then going to see a kind of the brief idea of the carbon cycling within this. So, we are going to discuss about the carbon cycling and biogeochemical processes of other nutrients maybe in some other class. So, these are the 4 major guilds, and as I said, there could be many other guilds which could be prevailed there based on the conditions which are prevailing there. So, this is one kind of an example. So, real situations might be more complicated. More zones can be defined because of the availability of the nutrients or the fluctuations within the physicochemical environment within this environment. The next point that I would like to discuss after this is the concept of microenvironment. Now, microbial habitats are as diverse as the microorganisms that live in them. So, we know that microorganisms are diverse, because just now we have seen that they could be photoautotroph, chemoautotroph, they could be heterotroph, they could be methane utilizing, they could be methane producing. There are many different types of microorganisms from their metabolism point of view, their taxonomy, their physiology. There are many ways that we can find out the diversity of microorganisms. But along with the diversity of the microorganisms, the habitats where they live are also diverse. For example, the normal the habitats that we see, the soil, rock, sediment, sand, water, different type of subsurface habitats, they are all representing the different type of habitats. There are extremophilic habitats also. Many environments, many habitats are extreme with respect to their temperature, with respect to their pH, with respect to their salinity. So, again within those environments we see a gradient of conditions prevailing. So, there are multiple habitats within each of these extremophilic habitats itself, and also there are some more interesting habitats like the surface associated. Surface means the plant and animal surface associated microorganisms. Those are habitats actually where, like the phylosphere, the leaf surface, where this is very special habitat on the plant leaves, where the microbial communities are present and they function only on the microbial plant leaves. The rhizospheric community or the root associated microbial community, these are externally associated communities. There are also intracellular habitats like endophytes, for example. So, they also represent habitats. So, what we see essentially that there are numerous habitats within our planet Earth or in the biosphere. So, along with microorganisms, the habitats are also significantly diverse. Now, within these habitats there are microenvironments. So, growth of microorganisms, as we have learned already, depend on nutrients and growth conditions available in their habitat. So, as we can see that with increasing nutrient availability, the microbial growth happens, and unless and until there are certain kind of other reasons, the growth will continue, and then it saturates. Obviously, at some point of time there will be limitation of some nutrients or competition among the species, and eventually the cell decay or cell death will begin. But before that, the supply of nutrients, the favorable growth condition will all favor the growth of cells present in any environment, and obviously their activities. Now, the habitat in which a microbial community resides. Now, a community is represented by numerous populations. So, within a habitat we will see numerous populations are there forming the community. The community which is present within a particular habitat is governed by the physical condition and also the chemical conditions, which are defined as the physico-chemical conditions, and together, as we have also learned earlier, that together these physical and chemical conditions, they determine the metabolic activities of the organisms which are present there. So, this physical condition and the chemical condition together forms the basis of the physico-chemical conditions, and the physico-chemical conditions basically drive the community composition and community function. It is not difficult to understand that the physico-chemical condition actually creates the strong pressure on the community structure, that what type of microorganisms will present there, because some of them might be very stressful, some of them might create a favorable condition for some organisms and unfavorable conditions from some other organisms. So, naturally they guide the community structure, and it is not only they guide the community structure alone, they also guide the community metabolic activities. So, the physico-chemical conditions actually control the metabolic activities of the community members. Now, you can easily take for an example the organic carbon rich environment like a paddy soil or a pond water or some similar environment where lot of organic carbon is available, compared to an oligotrophic environment where nutrients are very limiting, particularly the carbon is limiting, maybe phosphorus or nitrogen is limiting. So, if we look at the metabolic activities which are contributed by the microorganisms present over there, we see that the supply and the physico-chemical condition together they control the microbial activity significantly. Now, to understand this further, because the microorganisms they are very small in the order of few micron generally on the size scale, they directly experience only a local tiny environment. This small space is called the microenvironment, that is a few micrometer or few millimeter within their territory may be very significant for them, because they are themselves micron size. So, we will take one example after that. So, now the term microenvironment is used to describe the niche where a microorganism actually lives and metabolizes. So, where as we defined earlier the niche is the all the interactions the species member a particular species member they carry out among themselves and among their environment or among their abiotic counterpart to function in that environment, to live in that environment is the niche. So, the microenvironment is basically used to describe the niche within which a particular species function, but we should not get confused with the microenvironment that it is a kind of a synonymous to niche. It cannot be a synonymous, because in a microenvironment there could be many such species present, many niche. So, we will see that within a microenvironment a dozens of species or populations are there, and each of them is having their own niche into that. Now, a microbial niche is actually truly very small. As an analogy, we can take this example, that for a typical 3 micrometer rod shaped bacterium like this, a distance of 3 millimeters distance between this particular bacterium to another bacterium or some nutrient molecule which is available 3 millimeter away. It may be oxygen, it may be nitrate, it may be ammonia, it may be glucose or whatever nutrient, or maybe a cell who is producing something or who is consuming something. So, ultimately this bacterium is going to be affected by this bacterium or the nutrients which are present over there. So, the point is that a typical 3 micron cell will experience a huge distance related factor, because this 3 millimeter gap between the nutrient molecule or another species who might be producing something or consuming something on which this species has some interest, then this is equivalent to a distance that a human would experience over a length of 2 kilometers. Like this 3 millimeter distance could be equivalent to 2 kilometer distance for a human. So, just assume that you have a glass of milk 2 kilometer away. So, is it readily accessible? I do not think it is readily accessible. So, there may be a water tanker away from a person 2 kilometer away from the person. So, the person has to think that how I am going to access that glass of milk, or that it is just a representative, that is source of nutrition. So, it may be a glass of milk, or it may be a water tanker, or it may be a pond, or it may be some other nutrient resources, or maybe some other activities which are directly influencing or going to influence the human activity. So, it is so important. So, distance is going to a very important parameter in microbial physiology, microbial metabolism, and within ecosystem function. Now, within this 3 millimeter, which is 3 millimeter, for human it may be 2 kilometer, several micro environments may exist, and it is easy to understand. So, 2 kilometer for a human to a glass of milk distance might include many hurdles, many burdens in between. So, similarly, for a microbial cell, a 3 millimeter distance might have variations within its environment, physical or chemical parameters might vary, and each one of these micro environments. So, there could be numerous micro environments which are basically the alteration in their physical or chemical properties. So, each one of these micro environments will going to provide opportunities for different physiological types of microorganisms to thrive. Now, these micro environments will eventually will have different conditions in which other organisms will prevail. So, for example, if there is a bunch of organic carbon, or a kind of a huge deposit of organic carbon over here, and this bacterium is thinking that it is 3 millimeter away. So, I need to plan about accessing this nutrient. Either I can move, if I have a flagella, possibly I will move, or I will release some products, or I will see how I can access this nutrient or carbon source. But this bacteria will face numerous challenges or numerous favors, because in the intermediate space there will be many micro environments, and within each of the micro environments there will be numerous other bacteria present. All these bacteria would interact, some of them will surely interact with this carbon itself, and eventually the availability of the carbon or the overall physiological activity of this particular bacterium will be not only controlled by this carbon available over here, but also the micro environments and also the microorganisms who are living over there. Now, we can consider a soil particle. So, if we take a soil particle which is around 6 millimeter in its radius, we can see. Here we are representing a contour map. The contour map of oxygen concentration is actually plotted. This is an arbitrary plot, and there are actually methods by which micro sensors are available, through which one can measure the amount of oxygen present within a soil particle. Now, if we are able to measure the oxygen concentration within the soil particle, we will be able to see that the exterior of the particle will have almost the equal concentration of, or percentage level concentration, that is 21%, which is equivalent to the concentration of oxygen in the air. So, the external periphery of the soil particle will have almost like in equilibrium, it will have 21 or 20% oxygen. But as we enter inside the cell, or inside the soil particle, the oxygen level will be declining, and you can see that at the center of the soil particle the oxygen level will be almost 0, and it is going to represent a different zone of oxygen present within the soil particle. Now, in terms of oxygen relationship for microorganisms, each zone, so there are zones which are typically lack of oxygen, or just 1% oxygen, that is 5% oxygen, 10% oxygen, or there may be 21% oxygen. So, there are numerous oxygen enriched zones are there, and each of these zones are going to prefer or allow growth of certain microorganism within it. So, each zone can be considered as a micro environment. So, here in the picture we can clearly see there could be at least 1, 2, 3 micro environments or even more micro environments, because we have not plotted all the oxygen concentration zones over here. Now, this is actually going to affect the microorganisms present over in the soil particle. This is a story of a single soil particle. So, the particle has many micro environments now. The external or exterior most environment is the oxic environment, where we have almost the concentration of oxygen which is equivalent to the atmospheric oxygen, whereas the very interior part of the core of the soil particle is absolutely oxygen free, that is the anoxic environment. Now, with respect to microorganisms which are going to naturally occupy a soil particle and its different zone, so we will expect that within the core of the soil particle, which is basically oxygen free, we will have anaerobic microbes, whereas the oxic zone, which is the outer territory or the close vicinity to the outer territory, will have obligatory aerobic microbes, and the intermediate zone will have the microaerophilic microbes, because they are microaerophilic means they grow on or prefer to grow on low concentration of oxygen present over there, and facultatively aerobic microbes who can switch to strict anaerobic metabolism or anaerobic metabolism very well, and as well as they can perform the aerobic metabolism also, they will be spread across the soil particle. So, within the soil particle we can expect that there will be numerous or several micro environments, and each of these micro environments are occupied or populated by distinct set of populations within them, or distinct set of populations or community. They are actually, they will be forming community. So, the core of the soil particle will have a community, and the exterior part of the soil particle will have another community into that. Now, physicochemical conditions within this micro environment. So, there are several micro environments as we can identify. Now, these physicochemical conditions within this micro environment will vary temporarily and spatially. So, it is not a kind of a static phenomena that all the time the oxygen gradient will like this, because the soil particle may experience some kind of water logging at its top, and as soon as there is water logging, there may be decline in the dissolved oxygen concentration. So, this oxic zone might be converted to a micro suitable for microaerophilic organisms. So, this is not a static event. So, in any kind of particle or a small section of a microbial habitat, we can see that the distribution of microorganisms within each of these micro environments, or the dominance of any particular group, and over and above the activity of any type of microbial group present in these micro environments are subject to change, because this change is basically constrained by different kind of alteration in the physicochemical conditions. And also, it is not only this micro environment is working like this. So, within this micro environment we see that there are prime niche and fundamental niche. So, basically niche, as we defined earlier, where the microorganisms actually live and metabolize, and primary niche is also referred as the realized niche, that for every organism there will be at least one niche where it will be most successful. So, metabolically it will be in its peak, and it will be very successful in its performing in its activities. The organisms dominates the realized niche, but may also inhabit other niches. So, the dominant is the most preferable one, but any particular species can occupy other habitats also, and other niches it is less ecologically successful than in its realized niche. So, a microorganism or a particular species can occupy multiple niche obviously, but it will have a primary niche, and there will be many other niche which are the fundamental niches. So, together there could be other niches where the same organism, the same species may inhabit, may occupy, may grow, but remains less ecologically successful. For example, in several cases we have seen a bacterium like a species of Pseudoxanthomonas, or a species of Pseudomonas aeruginosa, or some kind of other species. So, these organisms often have been found to occupy several ecosystems, several habitats, but obviously all the habitats will have micro habitats, and in all the habitats they may not be very successful. In some niche they are going to be very successful, but there will be other niches where they are there, but they are not very successful. So, they may not be playing a very critical role or very important role within those environments. So, this part of my lecture is basically developed based on this material which can be followed, the block biology of microorganisms, and along with that the Prescott, Hartley and Klein microbiology and environmental microbiology from genomes to biogeochemistry. So, at the end we conclude this part of the lecture that interrelations among the populations that drive the ecosystem functions are discussed, concept of the guild is introduced, and microenvironment and how microbial community activity within a habitat may vary and ecosystem within the type of ecosystem, that is also highlighted. Thank you.